Hi, my name is Jenna, and today we'll be talking about the anatomy of the heart. So, the location of the heart is in the thoracic cavity, which is in the upper part of your body, and your heart sits between the lungs in a space known as the mediastinum. So, the heart has the apex, which is in the inferior part, which is like kind of the pointy part at the bottom here, and then our upper part and the broad superior surface is our base. So then our heart is also surrounded by a tough membrane, and that's called our pericardium. And then on the surface, we have these auricles, which are thin-walled structures that can expand when the atria are filled with blood. And we also have sulcus, which are fat-filled grooves where our coronary arteries will lie. And these are important for supplying blood to the myocardium. So now looking at internal structures of the heart, we have the atria, which are the receiving chambers. The right atrium is going to receive deoxygenated blood from the body, and then the left atrium is going to receive the oxygenated blood from the lungs. And then our ventricles are thicker walled, and these are the primary pumping chambers of the heart. And then we have these septums, which are partitions, and the interatrial septum breaks up the chambers, or the atriums, and then the interventricular septums breaks up the ventricles. So now we also have some more internal structures. We have 4 valves, which ensures unidirectional blood flow through the heart. So we have our AV valves here. So we have our tricuspid and our mitral. Our tricuspid has 3 leaflets, which I did not depict well here, and then our mitral valve has 2 leaflets. So they're both connected by the chordae tendineae, which connect to the papillary muscles, which control the opening and closing of the valve. Then we also have semilunar valves. So we have our pulmonary valve and our aortic valve, and these are 3 cusps of endothelium. And yeah. So now looking at the layers of the heart muscle. So our epicardium is actually the outermost layer, but it's also known as the visceral layer. And then we have our innermost layer, which is our endocardium. It is a simple squamous epithelium tissue with underlying areolar tissue. And then our thickest layer in our middle layer is the myocardium. And this consists of cardiac muscle tissue. It has our cardiac myocytes, and then we have the pericardium, and so this is a continuous membrane, and this is going to surround the whole heart. So the outermost part is the fibrous pericardium, and this is like tough connective tissue, and then we have the parietal and the visceral layer, and in between those 2 layers is the pericardial cavity. And this pericardial cavity, it's a fluid-producing membrane, and it has the pericardial fluid. And this fluid will actually help to cushion the heart and reduce friction. So now looking at the blood flow through the heart and the major vessels of the heart. So starting here, our deoxygenated blood comes from the body, from the lower and upper part of the upper part of the body, and it will flow into our superior and inferior vena cava. And this is flowing to the heart into the right atrium. And so this pressure will build up and open our tricuspid valve into the right ventricle, and then eventually the pressure will build up, opening the semilunar pulmonary valve, pushing our deoxygenated blood into our pulmonary trunk and arteries. So the pulmonary arteries will then carry this deoxygenated blood to the lungs, where that blood will then be oxygenated, and then that blood will flow back to the heart through the left pulmonary veins. And so those are the oxygenated blood from the lungs will flow into the left atrium, and this pressure will build up, opening the mitral valve, flowing that blood into the ventricle, and that pressure builds up, opening the semilunar aortic valve. And so our oxygenated blood then will travel up the ascending aorta, over the aortic arch here, and then into other arteries that branch off. So like we have our brachiocephalic artery, which flows to the right arm. We have our left common carotid artery, which flows to the neck and head region. And then we also have our left subclavian artery, which flows to the left arm. And then we have the descending aorta, which sends our oxygenated blood to the lower parts of the body. And so also on the surface of the heart, we have another system. So I'll also mention this, is we have multiple systems going on here. We have a pulmonary circuit. We have the blood flowing from lungs to heart, heart to lungs, and that's what's going on here and here. And we also have a systemic circuit where the blood is flowing from the body to the heart and then heart to the body. We also have another circuit, the coronary circuit, which is on the surface, which provides blood to the myocardium. And this is important because the myocardium houses all those conductive cells, which is necessary for keeping the heart pumping. So the coronary arteries, which I kind of drew out here, are supplying blood to the myocardium. And then we have coronary veins that are going to drain into the coronary sinus, and this returns blood back into the right atrium, and then the process continues. So now looking at the cardiac conduction system. So as we said before, the myocardium is a middle layer, and this is composed of contractile cells. Important thing to note is the difference between these two cells. The conductive cardiomyocytes function in spontaneously initiating and transmitting electrical signals, and so these are also our pacemaker cells. Then we also have the contractile cardiomyocytes, which are striated cardiac muscle cells that contract. They can be excited by conductive cardiomyocytes, and they can excite each other. Cardiac muscle tissue, so it's striated, it's mononucleate, involuntary, and autorhythmic. It has intercalated discs, which bind together by sarcolemmas from these adjacent cells. And these specific discs are going to consist of desmosomes, proteoglycans, tight junctions, and gap junctions, which all allow ions to pass between the cells and holds the cells together during this contraction. So the gap junctions are actually also going to allow action potentials to spread very quickly from cell to cell. Now looking at the cardiac conduction system components. So we have our sinoatrial node. So the SA node is where the normal rhythm is established, and it's the main pacemaker of the heart. It's depicted here, right there. And we also have our atrioventricular node. This is the AV node, and it's the second major structure in the conducting system. So it's critical that there's a pause before the AV node completely depolarizes and transmits the impulse of the to the atrioventricular bundle, and this will then travel that impulse down into the Purkinje fibers and stuff. So that allows the cardiomyocytes to complete their contraction that pumps blood into the ventricles before the impulse goes there. Then we also, which I briefly just mentioned, was the atrioventricular bundles, and they branch down into the apex of the heart and into the Purkinje fibers. And so these Purkinje fibers extend throughout the myocardium. They have a fast inherent conduction rate, and each ventricular muscle and can reach the ventricular muscle cells very rapidly. So now looking at the process of how this impulse travels. So we have our SA node here. So once this sends out that signal, it's going to, I forgot to draw it here, but it will, contract, send out that signal to the upper part of the atrium, and then this will then cause the AV node to hear that signal, and then that will then allow that impulse to travel down into the bundle and then into the here is when it's hearing that signal and traveling, and then that signal will then send to the cells of the ventricles, and then that's where that will contract. And so we have our left and right bundle branches here, and then these are the Purkinje fibers that are in here. That's our bundle of His in here. And then there's going to be a contraction here, and then this impulse travels, sends the blood, because it's contracting down here, up into the pulmonary artery or aortic valve, and so it's going to like push the pressure from the apex to the base of the heart. So the overall process is the SA node triggers that action potential. The action potential moves through the atria, and then that stimulates the contraction. The action potential depolarizes the cells of the AV node here, and then that action potential is propagated down the bundles and into the Purkinje fibers, causing contraction of the ventricles, and then this process then repeats. So looking at the action potential of a cardiac conductive cell, so this actually requires no stimulation by a neuron. So these sodium channels allow normal and slow influx of the sodium ions, and the membrane potential will increase, and then it will reach this point. And so this is where spontaneous depolarization will occur, and this is also known as pacemaker potential. So pacemaker potential is occurring here, and then our sodium channel prepotential, which is also the same thing as the pacemaker potential, influx occurs here, and then it reaches this threshold. And once this hits this threshold, fast calcium channels open, calcium is entering the cell, and it reaches this positive point, and it depolarizes until that positive voltage. And then the calcium close, and then potassium channels open. And this outflux of potassium repolarizations occurs until it hits back at that baseline. And then our calcium channels will close, and then our sodium channels open up again, and then this process occurs again. So calcium plays two major roles here. This is actually in our contractile cells here, though. Influx through calcium channels causes the plateau phase and refractory period that enables proper function of cardiac muscle. Again, that's in our contractile cells, and that's a separate action potential. And then also in calcium ions combine with the regulatory protein troponin and tropomyosin complex, and that allows myosin and actin interactions, which provides the power stroke of that contraction. And so we also have an important definition to know about is the overdrive suppression. So the whole idea is here. So if the SA node we know is the major pacemaker of the heart. So what happens if that fails? Well, we have our conductive cells take over that pacing, but how does that heart know which one should be pacing if it is working? So because the SA node beats at a faster rate than these backup pacemakers, that actually suppresses those pacemakers from firing, and that is what our overdrive suppression is. Looking at an EKG, it gives us the tracing of the electrical signal of the heart. So we have our P wave, which is the depolarization of the atria, and then we have our QRS complex here, which is the depolarization of the ventricles. Our repolarization of the atria is in here, but the ventricles have more of an input, so you will see that appear here. And then we have our T wave, which is the repolarization of the ventricles. An abnormal EKG could look something like this. This is a bradycardia, where the heart rate is less than 60, or here, where fibrillation occurs, and atrial fibrillation specifically, and there's a serious problem in the cardiac conduction system here. So now looking at the cardiac cycle. So we have our ventricle filling here, and so our ventricles are in late diastole, and our atria is still in diastole. So the blood pressure in the ventricles, or the vena cava and pulmonary veins, is greater than the pressure in the atria. So it's pushing that blood in, and then this is open and the ventricles are starting to fill, but the ventricles are still less pressure than the atria. And the semilunar valves are closed, and so as it moves over here, we have our blood filling in these ventricles. This is our atrial contraction / systole, and our ventricles are still in diastole. So the pressure or the volume that's in here is called our end diastolic volume, or our preload. And then the pressure is slowly increasing in here as the atria is contracting. So now in our isovolumic contraction / ventricle systole, our atrial is now in diastole. The blood is now in here, but it's still not at this point enough to open those semilunar valves. So as the ventricles now start to contract more and more, that pressure is going to push blood back, and that's going to cause these valves to close. And as these valves close, that's our first heart sound that we hear. So as these then the ventricles start contracting more, we're going to go into our ventricular ejection, and our atria is still in diastole. So our semilunar, because of that pressure of these ventricles contracting, our semilunar valves are now going to open, and then that volume pumped out is called our stroke volume. So that volume is going to push out into the aorta and pulmonary artery. And so now going into the isovolumic relaxation / ventricular diastole, and then our atria is still in diastole as well at this point. We have the blood flows back into the semilunars, and those close again. Same kind of idea here, and then that's our second heart sound. And then we also have an equation here that's important to know. Our N, to solve for our end systolic volume, it's our end diastolic volume minus our end stroke volume, or our stroke volume. And then so now we're going to briefly look at our cardiac output. So this is the measurement of blood pumped by each ventricle per minute. So we can find this by multiplying our stroke volume, which is the amount of blood pumped by each ventricle, times our heart rate, which is in beats per minute. So it's interesting because our stroke volume and our heart rate can be both impacted, and because of this, it will in fact affect the cardiac output. So our heart rate can be affected by autonomic intervention, hormones, fitness level, or age. And then we also have our stroke volume being affected by heart size, fitness level, gender, duration of a contraction, preload, and so forth. So then we can look at some practice problems here, and we can find our cardiac output given these values. So if we have our EDV and ESV, we can find our stroke volume. So we can do 120 minus 70, and that will give us 50. Sorry, my hand, it's really hard to type or write with this. And then we can multiply that by our heart rate, and then we will get 3,000. And because that's in milliliters, we want to get our answer in liters per minute. So we'll convert that 3,000 to liters, and we'll get 3 liters per minute. So now looking at the second question, if a patient has a regurgitation about 10 milliliters per blood, so meaning that blood is going backwards, so we're losing our forward stroke volume. We want to find the stroke volume, so what would that be? So using the same idea here, we have 130 milliliters, but then we also need to account for that 10 milliliters that is going backward. So we have to subtract 10, and then we'll subtract that 72 milliliters, and then we should get 48 milliliters. So thank you for watching. I hope this was helpful.